in video lecture 14, Energy Systems Part 2. So at this point, you guys have looked at Energy Systems Part 1, in which we looked at glycolysis and all of those steps, well, not every one in all of its gory detail, but basically what's happening in glycolysis. We've seen that little in-between step between glycolysis and the Krebs cycle, and we've seen what's happening in the Krebs cycle. So up to this point, we haven't actually seen a lot of ATP production yet. We've seen a lot of chemical reactions, and we've seen plenty of NADH molecules and FADH2 molecules being formed, but we have not seen a lot of ATP. So far we've only seen, we've netted 4 ATP molecules so far. So entering into this last part, the electron transport chain, we better see some ATP produced, otherwise this whole thing will have been for not much. So our objectives for this last section are to indicate where in the cell the electron transport chain takes place and the products of the electron transport chain. And then we're going to take a look at the basically the 3 macronutrients, because all along here, looking at ATP production, we've been looking at glucose as the fuel for making ATP. And so we'll take a look at other things that could happen to glucose in the body or in a cell. We'll also look at things that can happen with lipids and things that can happen with proteins. And in each of those, we'll take a look at what happens or how we make lipids into ATP and how we make proteins into ATP, because it's because we're not starting necessarily with a molecule of glucose like we've been looking at from the beginning. Okay, so that is what we're looking at in this video. So the electron transport chain, our third part of this process. So we know that so we know that the electron transport chain happens inside the mitochondria. Let's take a look at the mitochondria here, because the Krebs cycle happens in the mitochondria as well. So remember that glycolysis happens in the cytosol. So just outside in that fluid of the inside of the cell, that's where glycolysis happens. The pyruvate, that is our end product of glycolysis, is going to go through the membrane of the mitochondrion and pass through both the outer membrane, so see this outer membrane labeled here, and the inner membrane labeled right below it. So we can see there are 2 sets of membranes. There's an outer membrane, and then there's this inner series of membranes. So that pyruvate actually passes through both of these membranes into the matrix. Here's the matrix labeled, which is that inner part. So the other things we see inside this mitochondria are the purple squiggles are DNA. Remember that mitochondria contain their own DNA. So this molecule of DNA that we have in here is it codes for the information on how to make a mitochondrion. It doesn't have anything to do with our eye color or our hair color or, how to make mucus or any digestive enzyme or anything else in the whole entire body or that any cell might do. It only is the DNA on how to make this mitochondrion. And so they have their own DNA. It's called mitochondrial DNA, which is its own cool thing, because we get when at the beginning of life, when we start out, first we're a separate sperm cell and a separate egg cell. Those 2 cells come together. When those 2 cells come together, the sperm cell donates only DNA. The egg cell donates DNA and organelles. The egg cell is a very large cell compared to the sperm cell. The sperm cell is the smallest cell in the human body, and the egg cell is the largest cell in the human body. And so the egg cell has lots of cytosol and has organelles in it as well. And so, of course, some of the organelles that are in there are mitochondria, which means we all inherit our mitochondria exclusively from our mother, and which means we get our mitochondrial DNA, and how to make a mitochondria, we get that exclusively from down a maternal line. That's how that works. because the fraternal line does not donate any mitochondria to the newly fertilized egg. So anyway, so there are cool studies that can be done on mitochondrial DNA to kind of because there's not a lot of variation in mitochondrial DNA, and we can trace, like your family lineage around the planet by, subtle changes that have happened over millennia in mitochondrial DNA. So anyway, that's a whole nother story, but that's the thing. All right, where was I? Okay, the purple squiggles, that's mitochondrial DNA. The green balls, those are ribosomes. Those ribosomes are making proteins that are specific to the function of the mitochondrion. And those proteins we find some are embedded in that outer membrane. We don't really show them depicted here, but very importantly are the ones that are embedded in that inner membrane. So the proteins that are embedded in that inner membrane, we can see them like, they sort of look like, what are those things that you stick in oranges around Christmas time? Cloves, have you ever made oranges and you stab dried cloves into it? It smells so good and nice and Christmassy and stuff. That's sort of what it looks like here, I think. Anyway, so those proteins have been made by the ribosomes. They are embedded in that inner membrane, and those are the proteins that that's where the electron transport chain happens. Okay, is across those proteins. Okay, so what else do we need to say here? So let me, so in the notes we have sort of a general summary of the electron transport chain, and then we go through kind of, bullet point by bullet point, a little more specifically about what's happening. And then of course there is a video for you to watch that shows an animation of it happening, which is kind of nice to see because it makes it more clear. But let me give kind of a general rundown of what's happening here. So the electron transport chain, let me see, okay, water wheel, okay, I just had to remember what else we were looking at here. Let's look at this, the electron transport chain. So in this in this slide, we see 1, 2, 3, 4 larger blue globs, and then a fifth one that kind of is shaped like a not a beaker, but something like that you might see in a chemistry lab. So those 1, 2, 3, 4 blue globs are proteins, and they're numbered with Roman numerals 1, 2, 3, and 4. And then there's also a Q above the second one, and then in between the third and fourth one there's a site C, okay, which is cytochrome C, and the Q is coenzyme Q. So, and those are enzymes that facilitate this process. Those other 4 things are proteins that are also essentially facilitating this whole process. So what's happening in the electron transport chain, there's sort of 2 parts to it. The first part is these 4 proteins labeled Roman numerals 1, 2, 3, and 4. The second part is this other one that's labeled ATP synthase. So what happens in the first part is, remember all of those NADH molecules and the FADH2 molecules that were produced both in glycolysis and in the Krebs cycle. All of those guys show up here inside, they're in the matrix. So what we're looking at here as far as these membranes, right under the words electron transport chain, that membrane is the outer mitochondrial membrane. So if we go back to this diagram, it's that outer membrane that we see, and then that inner membrane, that's that second row of phospholipids that has the blue proteins embedded in there. Okay, so then inside of that we have what's called the matrix. That's where the Krebs cycle happened, and in between these 2 membranes is the intermembrane space, which is labeled right here, intermembrane space. So essentially what's happening in the electron transport chain is all those FADH molecules and FADH2 and the NADH molecules that were produced as products of the Krebs cycle, they are there in the matrix, and they start interacting with these 4 proteins. And essentially what they do is those electrons that they took on, remember they had been reduced in the process of the the redox reactions in the Krebs cycle. They had been reduced, which means they've gained electrons and they've increased their energy. So now they're going to pass these electrons in the form of hydrogen. So they're going to pass this through these proteins. So this NADH that we see on the left, it's going to give up its hydrogen and turn back into NAD plus, and then go back and be part of the Krebs cycle again. But those hydrogens that it passes through are going to separate, and the proton aspect of it is going to follow that red arrow up and end up in that intermembrane space. The electron aspect of it is going to get passed on to to that coenzyme Q. It's going to get passed on to that blue glob that has a Q on it. And continue to get passed on. So if we follow the yellow arrow across, we're following the line that the electrons take. So they're going to go through coenzyme Q. They're going to go through that next the number 3 protein there. They're going to go through that cytochrome C, the one that says site C, and they're going to go into the that last Roman numeral 4, that last protein. Once they go through that one, they actually then are, that's the end of the road for them, but they will pair up with oxygen and form water. So, and so that is what we need oxygen for. That's where oxygen comes into this whole thing. Remember in the very beginning we said this is why we need to breathe and it's why we need to eat. Here's the oxygen showing up. The oxygen is a hydrogen donor. And so water is constantly being made by hydrogen getting attached to oxygen. So water is a byproduct of this thing. Where did that hydrogen come from? Well, it comes from, so we also have this FADH2 that we see that's to the right a little bit of that NADH, and that's going into the second protein in this process and turning into FAD, which again is going to go back to the Krebs cycle and go and accept more electrons. But there are hydrogens that are being passed here that need to have a place to be received. And even though a whole lot of what's happening is protons, so hydrogen ions, are getting pumped into, in high concentration, getting pumped into that intermembrane space. That's one of the most important things that's happening here. But there are also hydrogen atoms that need to have a place to go, and so they get received by these oxygen molecules and turn into water. That's one of our byproducts, and we see that H2O that's that yellow arrow that's coming out of the number 4 protein is pointing to. That's the H2, the water that's being produced as a byproduct of this whole process. So there's that process that's happening. This passing of electrons, this pumping out of hydrogen ions. So all of these red arrows going up through these, the number 1, the number 3, and the number 4 proteins. In all of those places, we have hydrogen ions getting pumped into that intermembrane space. So we're ending up with a high concentration of hydrogen ions there. And that pumping process is being fueled by the passing of these electrons from one of these proteins to the next. The end story here, so that's all kind of the first part that's happening. And again, when you watch the animation, you'll see all that happening in action, and it looks kind of cool. So the end product, though, here is this ATP synthase. So with this high concentration of hydrogen ions outside the cell, or sorry, not outside the cell, in that intermembrane space, the only place that those hydrogen ions can go from there is directly through, so following that arrow that starts yellow but ends in a red tip pointing into the matrix. The hydrogen ions can flow through there. And as they flow through there, and they definitely want to flow through there because there's a high concentration of them in the intermembrane space, as they flow through, they literally turn that protein that's called ATP synthase. The turning of that ATP synthase takes ADP and attaches a phosphate group to it. That's our phosphorylation that we looked at when we were looking at the terms initially, and turns it into ATP. And so it's literally like a turning of a water wheel, like those old-fashioned, wheels, like at a sawmill or at a flour mill or something, where they would have a water wheel in a river, or they would flow water over it to turn this wheel, and that wheel would turn machinery inside the mill. So it's the exact same kind of idea where when these hydrogen ions flow in through this ATP synthase, it turns the ATP synthase and it makes it assemble this, it makes it assemble ATP. So we have to have ADP and we have to have phosphate in there for this to happen. And so once we have everything all lined up ready to go, then the hydrogen ions flow through there, the ATP synthase turns, and we start cranking out ATP. How much ATP do we crank out? We crank out 36 to 38 ATP molecules, well, 34 to 36 in this process right here. And then when we add those initial ATP molecules that we made from the Krebs cycle and from glycolysis, we net 36 to 38 ATP molecules. Okay, so here's our water wheel. Here's our summary. So I basically just, let me look at the notes. I just spoke through a summary. Let's assume I more or less said what's on all of these bulleted things, and then you can watch that animation and see it in process. Okay, but here's our net, our totals for the electron transport chain. So not our totals, but at least our reactants and our products. So we had, going into the electron transport chain, we had several NADH molecules, we had a couple of FADH2 molecules, and we needed water molecules in there, or sorry, not water, we needed oxygen molecules in there too, because remember the oxygen was going to be a hydrogen acceptor and turn into water. So that oxygen that we breathe, when it comes into cells, it goes into the mitochondria to be used for this. Our end products in the electron transport chain are NADH that will get recycled and turned back into NAD plus, that will get turned back into NADH in a future Krebs cycle. We have water. Oh, this doesn't say FAD, but there is also FAD that is an end product. We have water as an end product, and then we have our 32 to 34 ATP molecules. And so then this slide is also showing us that we made 2 ATP molecules in glycolysis, we made 2 ATP molecules in the Krebs cycle, and so that gives us a grand total of 36 to 38 ATP molecules from our single starting glucose molecule, which sounds like a big deal, and it is, but wait, there's more. So let's look at anything that a cell could do with glucose, and then we'll look at anything a cell could do with lipids and could do with proteins, and we will have an appreciation for other ways to make ATP as well. So, and we've mostly, I feel like we've looked at this in other classes, but let's just spell it out here. So when a glucose molecule comes into a cell, there are basically 5 things that could happen to it. One is ATP production. Cells definitely like to use glucose to make ATP. If the cell has needs for ATP, one of the first things it will prioritize glucose for is to use it to do glycolysis, to do Krebs cycle, to do the electron transport chain to make ATP. So ATP production happens with a lot of the glucose that we take in. Other things that happen with glucose that we take in are amino acid synthesis. So interestingly enough, if we're short on certain amino acids, we can make those amino acids out of glucose. So not every cell can do that. Liver cells are especially good at doing things like that. But we can take glucose, we can turn it into certain amino acids. We can't make all of our amino acids, but we can make some, and glucose is a good starting point to make some of our amino acids. So that's something that could happen to a glucose molecule. Glycogenesis is something that could happen to a glucose molecule, which is the making of glycogen. So remember that glycogen is our stored form of glucose. Skeletal muscle cells and liver cells store glycogen. And so if we have glucose around and our ATP needs are basically met and our glycogen stores are not full, then we might take that glucose, especially if we're a liver cell or a skeletal muscle cell, we might take that glucose and convert it into glycogen and just store it for later. So that's something that could happen to glucose. Another thing that could happen to glucose is lipogenesis. So this is the making of lipids. So if we have our energy needs met, we have our amino acid needs met, we have our glycogen stores full, then, and we still have plenty of glucose around, then we're going to start converting some of that glucose to lipids. Because we can only store so much in the form of glycogen, but we can almost endlessly store lipids. And so we'll convert glucose to lipids and store them away for future use. So that's the fourth thing that could happen to any given glucose molecule that comes into the body. And then if all of those things are happening and we still have lots of glucose around, we might pee out some glucose as well. So our kidneys will be like, hey, there's lots of glucose in this blood and we really don't need all of this, so we're actually just going to put some into the urine and get rid of it. So that's the other thing that could happen to glucose. So 5 different things that could happen to a glucose molecule once it finds its way into our cells. How about a triglyceride? So triglycerides, there are a few different things that could happen to triglycerides. One thing that we'll do pretty readily is store triglycerides because they're such a fabulous fuel source, which we'll see in a minute. So we may store them away for later, especially because as triglycerides are coming in, likely glucose is coming in at the same time, and we're going to prioritize using that glucose for ATP first. And so if there's lots of glucose, then we'll just store the triglycerides for later. And once the glucose has been run through and then we have ATP needs, then we'll pull the triglycerides out of storage. But triglycerides are also used for structure. So we've seen lots of triglycerides, well, not triglycerides, but we've seen phospholipids in cell membranes. So we can convert a triglyceride to a phospholipid if we need to do that to make cell membranes. We use triglycerides and other fats to make myelin in the nervous system. So there are various structural things that we'll use lipids for. So that's something else that could happen to triglycerides or happen to lipids when they come into the body. But also they can be used for ATP production. So how do we take a triglyceride and use it for ATP production? Well, remember that a triglyceride is made up of glycerol and 3 fatty acid chains. And so we can look at separately using the glycerol part of it and using the fatty acid part of it as energy sources. Plus, we can also look at using a byproduct of it also for ATP production. So let's see here. So where do we start? Let's start with the glycerol. So the glycerol, which is that backbone component of triglycerides, that glycerol molecule can be converted into that G3P molecule. So remember at the beginning of glycolysis, we took glucose, and in those first few steps, we converted glucose into that G3P molecule. Well, so we can take glycerol and convert it into the G3P molecule just exactly as if it had been glucose to start out with. So we can do that and then run that glycerol through the rest of glycolysis and end up with a pyruvate molecule and run that through the Krebs cycle and the electron transport chain and get ATP out of it. So from one glycerol molecule, we could yield like half the amount of ATP that we could yield from one glucose molecule, more or less, about half. So that's the glycerol molecule. The fatty acids, so we can see on this diagram, see the yellow fatty acids there on the right and the arrow going down to acetyl-CoA. So what happens with the fatty acids is we break them apart in pairs of 2 carbon atoms. And for each pair of 2 carbon atoms, we essentially can convert that directly into acetyl-CoA. So remember, acetyl-CoA is that step in between pyruvate from glycolysis and before the citrate of the Krebs cycle, we have that acetyl-CoA. So fatty acids can get broken apart in these pairs of 2 carbon atoms and turned into acetyl-CoA and fed right into the Krebs cycle. So if we recall the size of fatty acids can be 16 carbons long, they can be 18 carbons long, they can be 20 carbons long or longer. So for those 3 fatty acids of a triglyceride, say they're all 16 carbons long, so that would mean each of those 3 fatty acids could be divided into 8 sets of acetyl-CoA and fed into the Krebs cycle. So for longer fatty acids, if we count the glycerol molecule and all 3 fatty acids, if they're long fatty acids, we could get upwards of 300 300 ATP molecules per fatty acid. So, I mean, per triglyceride. So triglycerides are like the like home run of ATP production. Tons of potential energy is stored in fatty acids, or well, in triglycerides. So they're a fabulous fuel source and a really good thing to use to make ATP. So we can use the glycerol, we can use the fatty acids, but there's more. So we also, in the process of breaking down the triglycerides, there's a byproduct that's produced and liver cells can take those byproducts, and those byproducts are called ketones. They're specific molecules that are formed through the process of breaking down triglycerides. Ketones can themselves be used as fuel sources as well. So not all cells can do this. Brain cells can do it, heart cells can do it, kidney cells can do it. They can take ketones and feed those ketones into the Krebs cycle and use those to make ATP. So that's an additional fuel source that we can get from triglycerides. So they are a fabulous source of ATP for us. And okay, so our last one is what could possibly happen to a protein when proteins are taken into cells. So of course, first proteins are broken down into amino acids, and it's actually amino acids that come into cells. So the top priority for amino acids when they come into cells, or the top priority that cells have when they are taking on amino acids, is they're going to use amino acids to make their own new protein. So they have kind of a structural priority in cells. But if we are low on glucose, or we're low on triglycerides, or we have an overabundance of amino acids, then we can use amino acids to make ATP. So what does that look like? So remember that we have 20 different amino acids that make up all of our proteins, and those amino acids, each one has a slightly different structure. But what we're looking at here is the Krebs cycle, also called the citric acid cycle. It's actually turned on its side compared to how we were looking at it before. So the acetyl-CoA is all the way on the left, and the citrate then is going off of that and kind of going up and around that circle. But what we see in many of these boxes are different amino acids. So the box in the top left, we see leucine, lysine, phenylalanine, tryptophan, and tyrosine on the bottom. And so all of those amino acids we can turn directly into that acidoacetyl-CoA, which is the step before acetyl-CoA. On the bottom left, we have isoleucine, leucine, and tryptophan. So we've already seen leucine and tryptophan up above. But those amino acids we can turn directly into acetyl-CoA and feed all of those amino acids as acetyl-CoA right into the Krebs cycle and make ATP out of them. There are some amino acids, so if we go across the bottom, that middle square across the bottom, where we have alanine, cysteine, glycine, serine, and tryptophan. So you can see tryptophan, we can do various things with tryptophan. This is the third place we've seen it already. Those 5 we can turn directly into pyruvate, and then that pyruvate gets turned into acetyl-CoA and gets fed into the citric acid cycle, or the Krebs cycle, and so on. I'm not going to go through them all, but there are amino acids that we can see over on the right that get fed into the Krebs cycle at various places. So they become those various multi-carbon molecules along the way that we break down and harvest energy off of, and continue through moving around through the Krebs cycle. So there's lots that we can do with amino acids. We can essentially take our amino acids, turn them into slightly different molecules, and feed them into the Krebs cycle and get ATP out of them. So it's a little more work to do this with amino acids. It's fastest and easiest for our body to use, for our cells to use glucose to make ATP, and lipids and proteins take a little breakdown action to get them ready to make ATP, but that's okay, because especially for fatty acids, or for triglycerides I should say, they yield such a huge amount of ATP that it's really, really worth it. Whereas proteins, you can see proteins feeding them into, some of them feeding into kind of further into the citric acid cycle, we maybe aren't going to get quite as much energy out of them, but a lot of them, you know, we're going to get a lot because they're turning right into acetyl-CoA. So, so proteins can be a decent source of ATP for us as well. Okay, so that takes us to our objectives to check that we've done these, and I believe we have. So we've talked about where in the cell the electron transport chain takes place, which is in the mitochondria, and specifically in the, you know, along certain proteins in the inner membrane of the mitochondrion, and the products of the electron transport chain. So we had water as a product of the electron transport chain, and we had FAD molecules and FADH and NAD molecules as products, and we also had, of course, our main product that we're after, ATP. So lots of ATP is the product of the electron transport chain. And we talked about what could the 5 things that could happen to a glucose molecule when it comes into the body. We talked about a couple of things that could happen with lipids when they come into the body, and the various ways that we can take lipids and turn them into ATP. And we talked about what what our cells will do with proteins, converted, of course, first to amino acids, and what our cells will do with those, which is to use them to make proteins or use those amino acids to make ATP. So lots and lots of ways for us to make ATP, which is nice because we need a lot of it, and our body's good and very what's the word I'm looking for? Like, there are many ways that our body can make ATP. So, you know, we shouldn't ideally have a problem with it. Sometimes we do, because sometimes we don't have enough of the enzymes that we need to run that reaction. And so, of course, that reaction is going to stop wherever we run out of that enzyme. That's the end of the road. We're not going to make any more from there. Okay, so your homework is to answer these 2 quick questions. What happens to what happens to the process of cellular respiration if we're deficient in an enzyme? Oh my gosh, I was just saying this. That facilitates one of the reactions, or deficient in one of the interim storage molecules, such as NAD+. So remember, in the last lecture we talked about NAD+ is a derivative of a B vitamin. So if we're deficient in B vitamins and we don't have NAD+ to take on, to do that oxidation part, in other words, or not the oxidation, to be reduced to take on electrons and energy, what happens to the Krebs cycle then? That's the question that you're answering here in number 1. Number 2 is, what macronutrient yields the most ATP? So is it carbohydrates, is it lipids, or is it proteins? What macronutrient yields the most ATP? And I mean, I guess it could be partly debatable because it depends on what protein, but I think you know the answer because I talked about one of these 3 macronutrients as being a really fabulous supply of fuel for ATP production. Okay, so you're going to send me those answers, email them to me. That is it for this week, for week 7. Next week we are back at it, looking at more amazing things that cells do. So I hope this has been enjoyable for you, and I will see you guys next week. Have a great rest of your week.